explain this technically, but if you remember your high school biology, DNA makes RNA that makes proteins. And proteins are really what help us cell for a tissue or an organ function. Now, it turns out we had always thought that RNA was represented by what we call messenger RNA, which were long strands of RNA that were then read and turned into protein. But there are small pieces of RNA which are called microRNAs because they're only very, very short, but they can actually bind to these messenger RNAs that I was talking about and prevent them being turned into proteins. So in essence, in disease, they are inappropriately expressed. So you can have multiple copies of these small pieces of RNA that are then turning off proteins that should be there to maintain normal function. So ultimately what we're trying to do then is see if those small pieces of RNA that are inappropriately expressed can be detected in either blood, the CSF that Bob was talking about, and they represent the level of expression that these small pieces of RNA may be found in the actual brain tissue or the brain cancer.
Now, can I ask you a question? Because I think it's important for context, and I think I understand it. You're the expert. So we learned that there are about 30,000 genes, right, in the human. We used to think there were 100,000, but when they sequenced the genome, it turned out there were only about 30,000. And then you're telling me that these microRNAs exist. How many do you think there are?
So we have on our scientific advisory board some of the world's experts on microRNAs, and according to my SAP, it's 536.
So 500 of these short pieces, because
Yeah.
these are only 20 base pairs long.
That's correct.
And they regulate these 30,000 genes, 500 of them.
Yes, because one piece of microRNA can, because it's such a small piece, it can recognize a binding element within each messenger RNA. So one microRNA may recognize 50 messenger RNAs. So in essence, 500 microRNAs can then recognize 25,000 or whatever the right math is, messenger RNAs. So you can have this sort of amplification of the effect, so that one small piece of microRNA can influence not just one single messenger RNA, but multiple messenger